All right, guys, we're back with the last section of notes for Chapter 8 here, and probably in terms of material the College Board might cover, probably the most important one. It discusses the use of enzymes in chemical reactions. So we have discussed over the past couple of videos energy and energy dynamics and things of that nature, as well as metabolic pathways. And metabolic pathways are another way of saying chemical reactions that happen in your body. The chemical reactions that happen in your body would not happen at an efficient enough pace without the use of enzymes. So we are going to discuss today the use of enzymes, what enzymes are and how they work to facilitate these chemical reactions that we've spent 2 videos discussing already. Okay? So enzymes do speed up these chemical reactions, and they do so by lowering energy barriers that exist in these chemical reactions. And so that's what we're going to talk about here as kind of an introduction into what enzymes actually do. You may have heard of the term a catalyst before, and I know that that some of the material in just like regular chem or even honors chem or AP chem is something. I didn't want that one. Sorry. Is something that you guys may have heard of before, and but maybe in a different context. And so let me pop up here the there we go. I'm going to go ahead and draw on the screen real quick so you guys can see. We're going to clear that. I'm just going to make something up. Let's say A plus B equals C. Maybe this is just some really generic chemical reaction. And what you might know about a catalyst already is that it is some kind of agent. It doesn't always have to be a chemical agent, but some kind of agent that is added to a reaction that will help that reaction progress. And oftentimes in chemical reactions, you see that agent put above the arrow, right? So sometimes it might be like aluminum. I've seen aluminum there. I've seen heat above there. I've seen electricity up there, okay? Those are things that are oftentimes put above that arrow, indicating to you that a catalyst, which is one of these things that's added to the reaction, is being used to help that particular chemical reaction progress. Well, there are also biological catalysts known as enzymes. Okay, so enzymes are biological catalysts, and you could put the term enzyme there or a specific name of an enzyme right above that arrow, the same way that you would put some of these inorganic things here, like aluminum or heat or electricity. Okay, and so I'll show you an example of that, but what you do need to know is that enzymes are there as a type of catalyst, a biological one, that will help to speed up a reaction without being consumed itself. It works the same way as any of the other ones that I showed you, like aluminum or heat or electricity. The example that's given here in the last 2 bullet points discusses one that we've already seen before in Chapter 5. And so what you'll notice here, that we will have to apply some of the stuff that we've learned in Chapter 5 to this particular topic. And the bullet point here in number 3 says hydrolysis of sucrose can be done by the enzyme known as sucrase. And this is an example of an enzyme catalyzed reaction. And in fact, one of the things that you can kind of use as a rule of thumb in biology when you're looking at the names of molecules is that if the molecule name ends in ASE, it is oftentimes going to be an enzyme. Okay, it's one of those kind of cheat things that you see to yourself. Okay, if I see this thing sucrase, I know it's an enzyme, so I know it's a protein, and I know it catalyzes a specific reaction. And a lot of the enzymes that we see, that we look at, actually kind of are named for what they do. Okay, so sucrase is an enzyme that will help to break down sucrose. All right, so we see that reaction here in front of us. It's a little bit different, but similar to any normal chemical reaction you'd see like in a chemistry class. We see sucrose over here. Let me get the pointer out. We see sucrose over here, and they give you the structural formula of it as well, plus water. Then they give you the arrow. Then they tell you that the breakdown of this turns sucrose into glucose and fructose. Okay? But it also includes here over the arrow, just like I had said, is this catalyst known as sucrase. Okay? And so sucrase is added to this chemical reaction to make the transition between sucrose and glucose and fructose more smooth, okay? Sucrose, the breakdown of sucrose into glucose and fructose may happen anyway, okay? It may be spontaneous. I'm not really entirely sure, to be honest with you. But the enzyme sucrase is there in case it needs to be sped up, and it does need to be sped up, okay? So how does this work? How does the enzyme, the catalyst, the biological catalyst, work within these chemical reactions to make them happen more efficiently? Okay, that's the kind of the question we're going to answer here in the next couple of slides. Every chemical reaction between molecules involves bond breaking or bond forming, right? So we know that. And there is a certain amount of energy that is needed to start these chemical reactions, regardless of whether or not you're breaking bonds or forming bonds. There is always some kind of initial starter energy that is needed for this chemical reaction to occur, okay? And you have to overcome this initial energy kind of investment, if you want to call it that. And they call it activation energy, but we can call it energy investment. You always have to overcome this initial energy investment to actually make it so that the chemical reaction can occur. Whether it is bond breaking or bond forming, you still have this activation energy, okay? Now, oftentimes this activation energy comes in the form of heat from the surroundings, okay? So for human beings, the natural ambient heat that we produce, the natural metabolic heat that we produce, is enough to create a situation where the activation energy can kind of be overcome, okay? And so the same thing happens with a lot of other chemical reactions in nature as well, okay? But the activation energy is the energy that is needed to get the chemical reaction running. Now, this is a diagram that you'll probably see on the test, the AP test. I'm going to for sure put one on my test, and I can't remember an AP test where I haven't seen like an example question or a question on the actual test that doesn't have some kind of graph that looks similar to what you see in front of you, okay? But this graph is a graph that illustrates the amount of energy that the molecules have as well as the progress of the reaction, okay? It's very similar to, I just missed it. It's very similar to these graphs that you saw in the previous video, okay? Where you're looking at an exergonic reaction here and an endergonic reaction here. Very, very similar types of graphs. Now, obviously not the same, but very similar. And so what we're looking at here is we are looking at the reactants right here, which have a certain amount of energy associated with them, okay? Which is why they're kind of about halfway up on this energy graph, right? They have a certain amount of energy associated with them here. And then when those reactants undergo a chemical reaction and turn into the products, we see the amount of energy that the products has here, okay? And then we see the amount of free energy that's released when these reactants turn into these products, okay? That's what we see here with this kind of bracketed arrow, okay? So we have some energy related with the reactants, and then in this case a lower amount of energy associated with the products. And the amount of energy that was lost during that is depicted here. Okay, so what's this big bump? That big bump there is the activation energy. Okay, that big bump there illustrates the amount of energy that needs to be put into this reaction for it to actually work. Okay, and so we see these reactants with a certain amount of energy, but they require even more energy input to actually change into the products. Once they reach this area called the transition state, and this is important, the transition state illustrates a point at which the bonds that hold these reactants together become disrupted enough, I'm not going to say weak enough, but disrupted enough to the point where they will want to break. Okay, and we can see that they are becoming disrupted here by the wavy lines in between them. Okay, so that is akin to think about butter, right? Okay, so normally butter at relative room temperature, so about 70 degrees Fahrenheit, is going to be solid, right? But the minute you start to increase the heat or increase the energy on that stick of butter, you'll notice that the butter molecules start to liquefy. They start to melt, right? And what is occurring there? Pardon me. What is occurring there is that there's a disruption of the bonds that hold those butter molecules together, and eventually they will break and they'll slough apart. And the reason they were disrupted is because of the heat that you added. Okay, you can think of this as the exact same way. We add enough energy, oftentimes in the form of heat, where it will eventually disrupt these bonds to the point where they will want to break, very similar to how they would break when butter melts. And then when they do break, the new chemical reaction can occur, and B will make a bond with D, and A will make a bond with C, and then we get our products. Now these products, once they've been created, have a new amount of energy associated with them, which means they've lost some energy. At least in this case, this is an exothermic reaction, meaning that we have lost energy. Okay? There's been a certain amount of energy lost. And so sometimes that energy is lost to the environment as heat. Sometimes that energy can be harnessed by a cell to do work. Okay? So that's what we're seeing here with this energy diagram. Okay? So what we want to know is how do these enzymes lower that activation energy? Because if essentially that's what they do. They lower the amount of energy that is needed to kick off the reaction. That is how they facilitate the chemical reaction. So often I use this term that these enzymes facilitate chemical reactions, and that just means that they help them, right? So they help these chemical reactions by lowering the activation energy. Enzymes do not affect the change in free energy, okay? Instead, they make a reaction happen quicker, and they do so by lowering the activation energy. Okay, so you may also see a diagram that looks very similar to the one I'm going to show you here, or that I've shown you up on the board right here, minus the little colorful reactants and products. But this graph here shows you the course of the reaction with an enzyme. This is the red line that you see right here by the pointer that's moving. Okay. This is the line that shows you what happens when we add an enzyme. The black line is the course of the reaction without the enzyme. And what we see is that the reactants have the exact same amount of free energy to start, and the products have the exact same amount of energy at the end, which means the change in free energy is still unaffected. You're still going to get the same amount of free energy out of this reaction. But what has changed is this hump for the activation energy. You can see that it's much lower. Okay, it's almost half as high as the original amount. Okay, so basically what we have to do is put in less energy to start the reaction, and if that's the case, the reaction should happen quicker, right? We don't have to wait to build up this extra energy. We can kick off the reaction at a lower energy, which is going to take less time. Okay, so that's kind of how enzymes do what they do. Now, I've got another animation here that is good, so I'm going to replay it for you guys, and hopefully you can hear it. Very similar to what we've done in the past, okay?
Because of the specific fit between enzyme and substrate, if exergonic reactions occur spontaneously, what keeps molecules from breaking apart and cell chemistry from racing out of control? For any reaction to occur, even a downhill reaction, some energy must be added to get the reaction going. This energy is needed to break bonds in the reactant molecules. The energy needed to start a chemical reaction is called the energy of activation, E sub A. This required energy input represents a barrier that prevents even energy-releasing exergonic reactions from occurring without some added energy. How does a living cell overcome the energy barrier so that its metabolic reactions can occur quickly and precisely? A special kind of protein called an enzyme is the answer. An enzyme serves as a biological catalyst, increasing the rate of a reaction without being changed into a different molecule. An enzyme does not add energy to a reaction. Instead, it speeds up a reaction by lowering the energy barrier. An enzyme is very selective. Its three-dimensional shape allows it to act only on specific molecules referred to as the enzyme substrates. As the substrates bind to the enzyme's active site, they are held in a position that facilitates the reaction. This takes less activation energy than the unaided reaction. Products form and are released. The enzyme emerges unchanged from the reaction. Because of the specific fit between enzyme and substrate, each enzyme can catalyze only one kind of reaction involving specific substrates. Thousands of different enzymes may be required to carry out all of a cell's metabolic processes.
Okay, so that little video, that I think does a really good job of illustrating what you saw in this diagram here, okay, and what I had discussed. Now, the video had alluded to some specific vocabulary that relates to enzymes, which I have yet to cover, and kind of did that on purpose, but I want to make sure that you understand that the reactants, when we talk about the reactants in an enzyme catalyzed reaction, have a specific name. Scientists call these reactants the substrate, okay? And the substrate will fit into the active site of the enzyme, okay? And you saw that in the video as well. The little purple enzyme that we saw had a very specifically shaped, kind of recessed look to it. And the substrate, or the reactants, fit just right into that specific spot on the enzyme. That specific spot is called the active site, okay? So the enzyme binds to its substrate through the active site, forming something known as the enzyme substrate complex, okay? While the enzyme and substrate are bound, the catalytic action of the enzyme converts the substrate to product or products, okay? Now, one of the questions that is going to be, I think, important to understanding the enzyme as you should, is asking, why are enzymes so specific? Okay, why are enzymes so specific? And the answer to this is because of their three-dimensional shape. Okay, this specificity between the enzyme and the substrate is absolutely important. If you don't have this specificity, any enzyme can go run around and facilitate any chemical reaction. But when you have this specificity, now all of a sudden we can start to regulate the enzyme, okay? If you don't have this specificity, you get a situation where you have runaway chemical reactions, and runaway chemical reactions in a body is not a good look for the body, okay? You'll be making products that you don't need. You'll be wasting raw materials that you need for something else. You won't be making other things that you actually need. So this specificity in the enzyme is vitally important. And it is that specific because of its three-dimensional shape. Do you remember what gives this enzyme its three-dimensional shape? Well, pause the video and see if you can't figure it out. If you said the DNA, you were correct. DNA is responsible for the enzyme's three-dimensional shape, okay? Because the DNA codes for proteins, oftentimes, and enzymes are proteins. If there is a very specific change in the DNA in a very specific spot that codes for an enzyme protein, there's a really good chance that you could have an enzyme with an active site that is screwed up. Okay, even sometimes one small one small base pair change can create a three-dimensional structure in a protein that is completely off and different than what it should be. And if this happens with enzymes, sometimes that amino acid that is changed or off or different could be found in the active site. And if the active site has a different shape, it will not properly bind the substrate. So this active site three-dimensional shape matching up with the substrate is a vitally important interaction. Okay, never forget how important that is. Okay, and it plays a role in how the enzyme actually lowers the activation energy. So I've discussed here a couple of generalities. Number one, that enzymes help to facilitate a chemical reaction, which they do. They make them go quicker. They make chemical reactions go quicker without being used up themselves. And I've also said that they do this by lowering the activation energy. Those are the two big generalities that I've thrown at you right now. Okay, now I'm going to get into specifics. You might be asking, well, how does it lower the activation energy? Well, there's an answer to that question as well, and we're going to talk about it right here. Okay, this slide kind of gives you some words that describe it. I'm going to spend more time here on this picture instead. But this picture here illustrates and gives you some of the examples off to the side in number 3 as to how the enzyme will actually lower the activation energy. Okay, remember that the activation energy is the energy needed to make a chemical reaction happen, specifically an exothermic chemical reaction happen. And that energy can oftentimes be provided by the ambient temperature, the environmental temperatures, but sometimes that's just not enough, and you have to wait until the temperature gets just high enough, and sometimes that takes a while, right? Well, the enzyme is there to lower that activation energy. So let's just walk through this diagram little by little, and we'll hit on each one of these, and then we'll spend more time on another one, okay? So we'll start with number 6 here. I know it seems weird to start with number 6, but what we see at number 6 is an enzyme that is empty. It is ready to facilitate a chemical reaction. And we have to notice here that the active site is available, and we also have to notice that this active site contains a very specific complementary shape, okay? And it is shaped in a way that would be able to bind the substrates. And so we look up just right up north of it here. We see the substrates. There's a kind of a greenish-orange one and then a greenish-orange one. There's an orangish-red one and a green one. And if you look closely at the shapes of those substrates, they are complementary to the active site, okay? And so this active site has a shape that would fit these substrates. And so that's the first step, is that these substrates will enter the active site. Substrates will enter the active site. They will hopefully line up and fit right in the specific area of the active site that they need to be, which we see that they do. Okay, the red one fits up at the top and the green one fits up at the bottom. Okay, so at this point, what do we notice about the substrate? What has changed between this picture here and this picture here of that big blobby protein enzyme? That big blobby protein enzyme has changed from this picture here to this picture here. Pause the video and see if you can't figure it out. It has changed its shape. Okay, in the one in number 6, it looks like this, right? It looks like it's open. It looks like a Pac-Man that wants to eat some of those little, white dots or whatever they are. And then in picture number 2, where it shows the enzyme substrate complex, it looks like it's gone like this, and it's chomped down on them almost, right? That's a really good way of remembering what the enzyme does to help facilitate the chemical reaction, okay? So we read the picture here. It says substrates held in active site by weak interactions such as hydrogen bonds and ionic bonds. And so the substrates onto the side here, if you see where it interacts with the actual enzyme, there are going to be hydrogen bonds and ionic bonds that kind of hold those substrates to the enzyme so it can't like leave or something like that. It just holds those substrates in there so that they're nice and comfy. And on top of that, we see that the enzyme has changed shape and kind of clamped down on it a little bit. Okay, and that's going to become important because in number 3, that's where we start to talk about what the enzyme actually does to lower the activation energy. Okay? So it says there in number 3, the active site can lower the activation energy and speed up a reaction by doing one of these 4 things. It can act as a template for substrate orientation. Okay, number 1, we want to have these substrates oriented in just the right way so that they can form new chemical bonds more efficiently. So if for these 2 things to form chemical bonds, we want this edge right here of the red one to line up with that edge of the green one. Okay? And when they are lined up, they're going to form new chemicals, new products, much more efficiently. You don't have to be twisting them around and things like that. They're lined up exactly the way they have to be. Okay, so that specific site the specifically shaped active site, is going to make sure that anything that comes into that active site is going to be properly oriented to begin with. Okay. The second bullet point states that it stresses the substrate and stabilizes the transition state. Okay, so what this is talking about is this clamping motion. You can kind of think of it that way. If I were a strong man, like the steroid freak show that was in the beginning of the PowerPoint, you remember him? He probably can crush apples with his bare hands, right? He's got the forearms to do it. So think about an apple just sitting on your countertop at home. That apple is probably happy. It's probably not going to explode on its own, right? It's not going to turn into applesauce on its own, right? But what if enough stress was put on that apple, on the bonds of that apple? What if Strongman Freak Show in the first couple of slides of this video series took that apple in his giant hand and just started crushing it and putting as much pressure as he could on it? What's going to happen to the bonds of between some of those molecules of the apple? They're going to become stressed, right? And sometimes you can actually feel that. I might not be a strong man, but I know I can squeeze an apple to the point where I'm going to dent it. I might not be able to explode it, but I can sometimes feel the stress that apple is going to be put under, okay? And the same thing happens here. There is a change in the conformation of this enzyme that puts stress on the bonds of these substrates and causes them to become destabilized, okay? And it gets those bonds closer to the transition state of possibly breaking, okay? So there is some physical stressors in stretching and pulling and squeezing on the bonds of those substrates because of the enzyme that puts stress on the bonds to the point where they will want to break, okay? The third bullet point there states the active site provides a favorable microenvironment, okay? And this is where maybe I'll try my hand at drawing again. I don't know how it's going to go, but we'll pop this up here. Let's clear off this thing here. And I want to discuss what a microenvironment is. I'm going to get this out here. Okay, this is oh goodness, they don't give me the color I want. What's that? No. All right, we'll just do green. Okay, green. Here's a house. This is a log cabin in the snow, okay? And you can pretend that it's snowy, okay? And that's why I'm using blue. And I'm gonna draw this. This is like, we haven't experienced weather like this, and there's just snow everywhere. We haven't experienced weather like this yet. We're bound to at some point, and we're gonna have ice and wind and everything else, right? And this is a little log cabin in the middle of an Arctic forest, and it's snowing everywhere, and it's crazy. The wind is blowing and howling and blah, blah, blah. And imagine that this is a lighter shade of red or orange that's much more cozy and homey. Imagine that these windows, this house is not on fire, and it's not like a demon house. Imagine that this house has glowing lights inside, and it's got a nice warm fire inside, and you're walking along here as a person out in this blowing weather, and you're sad, okay, and you have offset eyes that are weird, but you want to go find some place that's a little bit more warm. Not a firehouse, but a house that has a nice cozy fire, and it lights up the doorway and the windows with this luminescent glow, and you walk in and you find that there's a bear skin rug on the thing and a nice recliner, and you've got plenty of wood for the night, and basically what I'm getting at here is that this cabin is akin to the active site of the enzyme. It is a very nice, favorable microenvironment inside this little log cabin that you see that I've drawn in front of you here. The weather is perfect. It's 80 degrees. There's a nice fire. You're not uncomfortable. Everything is just perfect inside of this log cabin while the winter storm rages around you on the outside. That is kind of like what is going on in like a cell, right? It could be just crappy in the cytoplasm, where this chemical reaction between green and red here just doesn't happen because the environment's not right for it, okay? But inside of this little active site that we see here, it's perfect. The pH is wonderful. Okay, it's a nice pH of about 7. The temperature is great. It's about 21 degrees Celsius. It's a great pH. The salinity levels are perfect. They're great. And this chemical reaction can happen like it was supposed to, because everything inside of this favorable microenvironment is happy and cozy for these guys to do what they have to do, okay? And obviously that microenvironment might be different for every different type of chemical reaction that is facilitated, okay? And then finally, sometimes the amino acids that are located on the edges of the active site can participate directly in the reaction. So they can have some kind of participation between green and red to help that reaction occur more quickly. Okay? So as a result, all of these things added up, all of these nice things taken into account, we take the green thing and the red thing and we combine them in a certain way, and all of a sudden they make the blue thing and the yellow thing, which are the products. And then the products are released from the active site and the enzyme is ready to do it again. So that is how the enzyme helps to lower the activation energy. Yes, it's kind of complicated, but and there's probably even more detail to that, but each enzyme is going to work specifically with its substrates to do any number of these things here to lower that activation energy. So if you really wanted more detail, you'd have to study a specific enzyme substrate complex to understand it a bit better. Okay. Now, I mentioned the effects of the environment on chemical reactions, and the effects of the environment also need to be taken into account when discussing the enzyme, because the environment can drastically affect how the enzyme actually works. Now, we've discussed this a little bit in Chapter 5, right, already. We've talked about what effect temperature and pH have on proteins, and the exact same discussion can be had here. In fact, I'm not going to spend a whole lot of time on it because we've already talked about it, right? We've talked about if the temperature becomes too high with an enzyme or a protein, that it can denature. It can just break apart the hydrogen bonds, the ionic bonds, and things like that are keeping the structure of that molecule together. At the same time, if the temperature is too low, maybe you don't have fast enough molecular movement for that enzyme to really do its job. Okay, that's going to be an issue as well. We look at pH. Okay, pH of the environment can have an effect on the enzyme as well. Too high or too low of a pH can disrupt the hydrogen bonding of that enzyme structure and denature it. Okay, so what you'll find is that each enzyme does have an optimal temperature or pH or even salinity that it works best at. And you'll see, I think I'm planning on, or I have in the past, put a question on the test that has almost this exact diagram. Okay, so you might want to take some time and just go back and review this exact diagram. Okay, but this diagram here shows two different enzymes, and it shows the enzymes in different temperatures and different pHs. So the graph at the top shows enzymes throughout different temperature ranges, and the first one up here says the optimal temperature for typical human enzymes, and you can see that they measure this by the rate of reaction, how quickly the reaction happens. So the most efficient enzymes in the most efficient environments yield a rate of reaction that is at the peak here, that is fastest, okay? And we see here that the optimal temperature range for human enzymes is somewhere in the neighborhood of 37 degrees Celsius, which is about body temperature, 98.6 degrees or something like that, okay? Whereas you have some bacteria that actually have enzymes that will need to work at a higher optimal temperature because they're heat-tolerant bacteria. So whatever puts their enzymes together, I don't know what the structure of those enzymes is, but their enzymes work best at about 79 degrees Celsius. Okay, that's really high. Okay, that's probably in the neighborhood of 150 degrees. We would die. We would die. Our enzymes break apart. We'd be dead real quick if we tried to work at that particular temperature. Okay, but for these guys, that's where they work the best at. Okay, there's even, if we look down here for pH, there's different types of enzymes in our body that work at different pHs. So for instance, this one here, the enzyme known as pepsin. The enzyme known as pepsin works at an optimal temperature of about 2, just under 2. I bet you wonder why, because it's in the stomach. The stomach's pH is about 2, is very acidic, all right? Whereas this other one called trypsin is an intestinal enzyme, and it works at best at a pH of 8, okay, a little bit higher than neutral. So there's different optimal ranges of these enzymes and which temperatures and pHs they actually work. All right, Section 5, we're almost done here. Section 5 discusses the regulation of enzyme activity. If we could not regulate the workings of our enzymes, we would have, as it states there, chemical chaos. You'd have some pathways that would just keep going over and over and over again. You'd have some pathways that just wouldn't go fast enough. And you would not have anything that resembled life. It would just be chaos, okay? And so to regulate metabolic pathways, the cell switches on or off genes that encode specific enzymes, and in some ways those enzymes can be regulated themselves once they're already created. And so that's what we're going to talk about here. The first way that we're going to talk about is enzyme inhibitors, okay? So the enzyme inhibitors work on enzymes that are already around, okay? We have two different types of inhibitors. We have competitive inhibitors and non-competitive inhibitors. And so they work, the ultimate effect is that they both inhibit the functioning of the enzyme. They just do it in different ways. Competitive inhibitors will look for or compete with, that's where you get the term competitive, right? Compete with the normal substrate for action, if you will, in the active site. Okay, so if we look at this example right up here, this is the normal binding of a substrate, here shown in turquoise blue, blue-green, to this enzyme, which is purple. Okay, you see that it's got this specific shape that fits in this specific active site. And if it does that, it will have some kind of chemical reaction with it. It'll probably break apart into two things or whatever, okay? Whatever it is, a normal chemical reaction will happen when we're looking at this particular picture. Now, if we use a competitive inhibitor, for instance, a competitive inhibitor is something that, some kind of molecule that has a similarly shaped, is similarly shaped to the active site. Okay, so we see here that the inhibitor is not the exact shape, but it's close enough to fit into the active site. And what it does is it gets in the way of the normal binding of the substrate. So it blocks it. It's competing for time in the active site. And when it wins time in the active site, then we have the substrate bounce out and doesn't get to do what it's normally supposed to do. Okay? Non-competitive inhibitors work a little bit differently. The effect is still the same. The substrate is not going to be able to enter the active site, but the way in which this occurs is different. This orange thing down here is a non-competitive inhibitor, okay? And if you notice that there's a part in this normal enzyme down at the bottom, it's almost like, it looks like a secondary active site, and things can bind in here, all right? And what we see is that the non-competitive inhibitor can actually bind to this spot on the enzyme. And when it does, for whatever reason, there's some kind of conformational change in the actual active site. These little horns that kind of stick up at the top normally and allow that substrate to enter, we see that when the non-competitive inhibitor enters its active site, it will cause these horns to kind of close, almost like closing the gate. And so when that gate is closed, it changes the shape of the active site, making it so that the actual substrate can't bind and then bounces out, okay? So the chemical reaction won't happen. So we're going to talk about a specific example of this one as we work our way through, okay? There is also another way, a couple of other ways that we can regulate enzymes through something known as allosteric regulation. And that's a term used to describe cases where a protein's function at one site is affected by binding of certain molecules at a completely different site. So these allosteric regulations can either inhibit or even sometimes stimulate the enzyme's activity. Okay? So in enzymes that work through allosteric activation or regulation, the enzyme has active and inactive forms, meaning at some points in time during its life it's got this form, this shape that makes it so it's active and it can facilitate chemical reactions, and that other times during the life of the enzyme it has this inactive form, which means that it's kind of closed and it's not going to be able to facilitate these chemical reactions. Okay? So allosteric regulation acts through activators, which you see right here. Activators, which are specific molecules that will work to stabilize the active form of the enzyme. But they also work through inhibitors, which will help stabilize the inactive form. This is easier seen with a picture here, okay? So these, some of these allosterically regulated enzymes will actually oscillate between their active and inactive forms. If you have studied a little bit of organic chemistry, and I don't know which chemistry teachers still do this, but when you get into college, you'll understand that there are some types of molecules that can actually oscillate their forms, meaning they're constantly changing their forms. Okay, oftentimes we see this in organic ring structures where like a double bond will like rotate. Okay, it'll oscillate between certain forms, and so that's kind of what's happening here with the enzyme. The enzyme that we see can oscillate between this active form, where if we look at these active sites, they're open and ready to receive some kind of substrate. There's 4 of them in this particular little thing. That doesn't necessarily matter. But there's 4 different active sites here that are ready to receive substrate and make a chemical reaction occur. Okay? Whereas the non-active or inactive form looks like those active sites have the gates closed, very similar to what we had just seen. Now, nothing can really happen when this thing's oscillating back and forth. It does it very, very quickly, and very rarely are you going to get a substrate that bounces in there at just the right time. But these things are oscillating back and forth, back and forth, back and forth. So we need molecules to stabilize it in one form. And one of the molecules that we can use to stabilize it into the active form is known as the activator. Okay? And so the activator comes along and it sticks right in here. And we see that has happened here. And what that's going to do is going to basically lodge it. Okay? It's not going to be doing this anymore. It's going to go stick it there. And it just stops in that active conformation. Instead of bouncing back and forth, it'll just go, okay, we're ready to go. And then things can, substrates can start entering the active site here. Chemical reactions can become facilitated, and this thing's doing its job. It's on, okay? Whereas we can also use inhibitors. If we don't need the chemical product that is the result of this enzyme's activation, then we can kind of lodge this enzyme into its inactive form using this inhibitor molecule. And the inhibitor molecule is going to see this oscillating molecule and stick it in its inactive form, which is like closed, okay? Here the active sites are open for the activator, and here the active sites are closed for the inhibitor, and it locks it into place as we see right here. So as this thing oscillates, this little molecule finds its way right in here, whoosh, sticks right in there, and it locks this inactive form so that nothing can actually be created, okay? So that's how allosteric regulation happens. Now, this can be used in feedback inhibition, and this kind of goes back to chapter 40 stuff. We talked about feedback a little bit, right? Where in this case, this is a very specific example of negative feedback where the end product of a particular metabolic pathway will shut down the pathway, and it will do so through inhibitions that we've just talked about in the previous 2 slides, okay? And so this type of negative feedback prevents the cell from wasting raw chemical resources that it doesn't need to be wasting, okay? So let's look at this specific, I'm going to get out my drawing utensil here. This is the last slide, so you'll only have to bear with me for a little bit longer. All right, so I'm going to move this closer to me real quick. All right, so what we see here is an enzyme that they call threonine deaminase. Okay, we know it's an enzyme because it ends in ase, and we don't really care what it does at this point. Okay, but what we do care is that threonine deaminase is going to bind to something called threonine. Okay, threonine is the substrate in this case. Okay, and when threonine binds to the active site of threonine deaminase, we will start to initiate a chemical pathway. Okay, so whatever happens here you're going to get this intermediate, and then you're going to have another enzyme activate, and then you're going to get another product which interacts with another enzyme, which interacts with another one. And so this is what we call a metabolic pathway. There's 4 different chemical reactions that happen after the very first one does. So by the time we get through all of these we'll call that a metabolic pathway. By the time we get done with all of these chemical reactions in this particular metabolic pathway, we end up with the end product called isoleucine. Okay, and this is isoleucine is an amino acid. Okay, now if this continues to happen over and over again, we're going to get a lot of product. Okay, if this pathway stays on, we're going to get lots of isoleucine, right? Just naturally, right. If we keep this thing on, it's just going to keep making it over and over again and over and over again. And ultimately, we're going to end up with lots of isoleucine. And that's okay because our body, for whatever reason, needs isoleucine, so it wants to continue to make it over and over again, okay? But the body only needs so much. It doesn't need infinity isoleucine. It only needs an X amount of isoleucine. And we want to be able to shut this off. Otherwise, it'll just continue to make it until we run out of resources and we die because we've used all our resources making isoleucine that we don't need. So what isoleucine is going to do is going to feed back into the system. Okay, so here is that same enzyme that we started with. It's right here. It's got this initial active site that we see. This is where threonine binds to. This is the original active site. But it's also got this other active site down here, which is used for the non-competitive inhibitor, right? And so what actually happens, I don't like the sequencing of their drawing here, but that's okay. I didn't make it, so I can't complain too much. Isoleucine is going to act as the non-competitive inhibitor for the original enzyme. Okay, isn't that interesting? This isoleucine is specifically shaped so that it will fit right in here to this part of this kind of secondary active site. Okay, and that's what we see taking place in this picture right here. That's what we see happening right next to the blue check mark, is that the isoleucine binds to this allosteric, non-competitive inhibitory site. Okay, and when it does that, it takes these horns, which are normally up like this, receiving threonine, and it closes them, which is what we see in this picture. It closes them off. So the active site can no longer bind threonine the way it normally would right here, okay? So if that active site can no longer bind threonine, we effectively shut off this step right here, and if we shut off this first step, it's going to shut off this step, which will shut off this step, and everything else, kind of like this crazy cascade of shutoffs. And so this is what happens. We get that isoleucine that goes to the non-competitive site. It closes the active site that binds threonine in the initial step, and then it shuts down the entire pathway as a result. Kind of a really specific look into a really specific negative feedback loop using enzymes. It's pretty cool, in my opinion. Okay, and so the reason this works is because there's so much of this isoleucine that we see down here as the product that it will inevitably run into this allosteric site that we see at the bottom, okay? And it will then stabilize this kind of inactive site, this inactive form of the enzyme, and shut everything down. Kind of cool, right? So you have now been exposed to a very quick crash course in enzyme activity and the importance of enzymes, and I really do hope that you can come up with some good questions to ask in class, or ask some questions about anything in terms of clarification, okay? So I will talk to you guys shortly, and enjoy the rest of your day.